Hello and welcome to another edition of the Omics Cast, where I talk about uh, genomics and omics in general technology. My name is Roberto Vilella. I got a PhD in bioinformatics from the University of Barcelona. I worked for a bunch of years at the European Bioinformatics Institute in Cambridge, United Kingdom. And I also worked in the past for Illumina, and I am currently the head of precision bioinformatics at Cambridge Epigenetics. And today I'm going to talk about the Illumina technology in terms of their flow cells. In this presentation, Illumina pattern flow cells, implications for omics assays. Illumina traditionally has had one type of flow cell called random clustering. It was a kind of a glass flow cell with some molecules attached to it that would allow for the molecules of your sample to hybridize attached to it. And these molecules, they would land randomly on the flow cell in the process that was called seeding. And then after that process was complete by passing through some liquid through the flow cell where you had your starting material, then they would apply another, it's still happening today for some of the machines, they would apply another set of reagents and then grow each of the molecules that had landed randomly on the flow cell. And this would produce what's traditionally called random clustering. These work very well for many of the machines that Illumina is still selling, like the NovaSeq machine, the old HiSeq 2500s, the MiSeq, and it allowed Illumina to dominate the market share. Illumina then later on came up with this pattern flow cell, which is nanowells that are positioned at a specific place with a specific density in the slide in the flow cell. And then this works differently from the previous system. Here the molecules can land and then immediately grow into the nanowell. And then molecules cannot land and grow outside of the nanowells. And then this pattern flow cell technology has allowed the company Illumina to increase the density and the number of clusters that are produced in a sequencing run and produce more data. Something that came up when, as evidence, when Illumina started deploying the pattern flow cell technology, mainly on the HiSeq 4000, later on with the NovaSeq, is something that's been dubbed or called index switching or index hopping. And there was some evidence of it in the literature, and then Illumina came out and provided with more information about how this was happening, who was affected, and what type of preps were affected by it, in a white paper that they called Effects of Index Misassignment on Multiplexing Downstream Analysis. And then came up with a remediation and a set of indexes that you could use to make this problem essentially disappear. So index hopping from the Illumina white paper, it's essentially what happens when you have molecules being processed that are processed with a different tag that they should, right? It causes incorrect assignment of sequencing reads and then may lead to misalignment of reads or incorrect assumption downstream analysis. The HiSeq machine was the first machine for Illumina to deploy a pattern flow cell, and its application initially was only targeted for whole genome sequencing. And then the most common type of application is that you would put one sample on each of the lanes of HiSeq to get roughly a 30x coverage human genome. So it did not require multiplexing. Essentially, it was just a matter of putting your one sample in that lane, and that lane would contain all the reads from your sample, not from other samples. The HiSeq 4000 was the second machine with pattern flow cell technology. It was essentially the same technology as the HiSeq, lower spec throughput, but allowing for multiplexing so that people could do exomes, RNA-seq, etc. And so the HiSeq 4000 was the first multiplexing, first time the multiplexing was used on pattern flow cells. What was discovered is that in the process of the molecules landing on a nanowell, as seen here, they have all these primers where they can hybridize. They would land and grow and produce copies. There is a small probability of one of these copies or the original copy detaching itself from the nanowell it started growing into and then landing on a different nanowell. And when it landed, it could cause problems like duplicates and other problems. The pattern flow cell versus random clustering had more of this index hopping percentage, so, and more so when you had PCR-free applications. PCR-free is great, is essentially a method of preparing your library with Illumina adapters so that you can put it through the sequencer straight away without having to amplify the sample. And it produces high quality data. It doesn't produce any errors caused by the polymerase through the amplification process. So it's basically a way of saying, this is my sample, adapt it, put it through the sequencer, and then get the immediate result from the sequencer at a very high fidelity with very high accuracy. If you have to amplify your sample, either because you only have a few nanograms of it or for whatever reason, then the end product of it will be slightly noisier. But when it comes to index hopping, it didn't cause that much index hopping in comparison. And as you can see, in the pattern flow cell, it's roughly about 5 to 10 times a larger problem for the compared to the non-pattern flow cell. So index switching happens where the original molecule green-red switches into green-yellow, and where you have a pull here mixed with the exam reagents, the original molecule can switch into the incorrect index, I7 and I5 index, when it's copied through. So if you rely on the indexes to tell you which of your samples is which, then when this index swapping happens, you basically have misassigned that molecule from then onwards. And then the solution is to restrict the indexing combination so that you can find out when this error has happened twice in one single molecule rather than only once. Single indexing reads work like this. Essentially, there's a part which is your DNA insert. That's what the biological sample contains. And then there are a series of additions that Illumina uses to basically classify your molecule, your set of molecules in a multiplex sample. This works great because it allows you to tailor your sequencing run, put as many samples as you need to get the expected throughput rather than having to have too many reads from a single sample. In dual indexing, you essentially do the same, but in the middle of the run, you read indexes from both ends of the molecule of the DNA insert. And then you end up having two k-mers, two 6 bases or 8 bases nucleotides to tell you exactly what your molecule is, where it belongs in the multiplex to run. So if you combine the probability of both indexes getting it wrong with dual indexing, the probability of index hopping goes quickly to 0, because if you use indexes that are different enough between I5 and I7, the probability, as you see in gray here, of index hopping of molecules of these combinations ends up being almost null. So Illumina came up with a series of best practices, and then later on they themselves started selling these dual indexes that would resolve the problem, and they weren't in any way more expensive or anything else compared to the previous indexing systems. And here are different combinations where you can do the dual indexing. So the dual indexing basically got rid of this problem, and even though it was a problem, for a short period of time that people noted in their experiments, there is now a very good solution to get rid of this issue. It lowers the contamination. It allows you to do the sequencing runs without any misassignment of the samples. And the issue was more prominent with PCR-free preps, but if you manage to get rid of the issue of index swapping and still use PCR-free, then you get very high quality data at the multiplexing rate that you want. And then there's another aspect here, which is it has been found recently with relation to pattern flow cells versus non-pattern flow cells. And this is completely different from index sampling, completely different from anything else. It's for a specific application, which is metagenomics, or specifically meta-barcoding, where a series of samples is sequenced, but we don't know what species those molecules belong to. And then we use the readout of each of the sequences to assign it to a species, known or not known, compared to a database, for example. So a published study in Nature group claims that the pattern flow cell technology results in detection of higher diversity values in samples compared to the MiSeq. So the pattern flow cell of the NovaSeq works differently to the non-pattern flow cell in terms of knowing where the nanowells are in the flow cell and imaging them, expecting the wells to be in a specific place, compared to the random clustering flow cells where you really don't know where the clusters will grow, and so you have to first take a few pictures and find out where the clusters are to then determine the boundaries of each cluster. So this would be an alleged issue for metabarcoding applications, where if you use non-pattern flow cells, you discover less of the diversity existing in that sample than if you use pattern flow cells, for example, the NovaSeq. And immediately you can think, well, if you compare what you get out of a MiSeq, which is a very small machine, low throughput machine, to the NovaSeq, are you telling me that you get more information from the NovaSeq? Well, maybe that's because you're sequencing much more in the NovaSeq. Well, the authors in this experiment, they tried it the hard way. They basically got as much sequence out of MiSeq runs than NovaSeq runs, and then compared apples to apples these results, and then concluded that the NovaSeq or any pattern flow cell Illumina machine, NovaSeq, HiSeq 4000, or the smallest of them, the HiSeq 100 machine, is better at detecting diversity than any of the non-pattern flow cells for the same throughput. And so the NovaSeq found 40% more metazoan families in a specific experiment than the MiSeq, including some of interest such as marine animals and bony fish. So the real-world implications of this finding are significant. Essentially, if you want to detect or catalog rare or endangered species from your meta-barcoding application, it's better that you use pattern flow cells like the NovaSeq. If you continue using the MiSeq, you're likely to miss some of these rare molecules that are there that you wouldn't find otherwise. So the NovaSeq, the authors conclude, is superior in finding that biodiversity, in finding this diversity in the molecules, the DNA molecules, for environmental meta-barcoding studies, especially when you want to find samples that are rare, molecules belonging to species or variants that are rare. And then they say, not everybody can afford a NovaSeq. The MiSeq has a 600 cycle kit that is quite useful for some applications, including metagenomics. And up until recently, the NovaSeq or any of the pattern flow cell machines could only achieve a 2x150, so 300 cycles, half the number of cycles of this MiSeq kit. But Illumina recently has announced and made available a 2x250 kit, both for the NovaSeq and the HiSeq 100. So now they're much closer to the levels of read length that you could get from a MiSeq. So in conclusion, the pattern flow cell technologies are different, fundamentally different to the random clustering flow cell technology that Illumina has. And this has implications that need to be understood. And then when you're running your application, you have to know where you are and to extract the best value for the different types of platforms that Illumina sells. And it's not that the pattern flow cells are bad. And as you can see, there are cases where they're much better at doing something that people were attempting to do before on other machines. And this has been the presentation for this Omics Cast episode. I hope that you enjoyed it, and I hope to be doing more of this in the near future. Thank you very much.